Alright, so this is our stop motion video, and it's by Bishesh,
Mrugank,
Bryan Wynn, and
Jason Garza.
so what we're going to do is define and explain the stages of mitosis. So we're going to start our video, and the first one is interphase. And interphase is actually not a part of mitosis, and interphase of the eukaryote cell can be subdivided into the following three phases. The first one is growth phase 1, and during this phase the cell grows rapidly while performing routine metabolic processes. And stage 2 is synthesis phase, and during this phase the cell's DNA is copied in the process of DNA replication. And stage 3 is the growth phase. During this phase the cell makes final preparations to divide. For example, it makes additional proteins and organelles. So this is our interphase.
Okay, next I'm going to talk about prophase, which is the first and the longest phase of mitosis. And during this phase, chromatin, which is uncoiled DNA, condenses into chromosomes, and the nuclear envelope, or the membrane, breaks down. So prophase is the first and longest phase in mitosis, and during this phase, chromatin, which is uncoiled DNA, it breaks down into it breaks the membrane into chromosomes, and the nuclear envelope, or the membrane, breaks down. Okay. So this is how the prophase process happens. In animal cells, the centrioles near the nucleus begins to separate and move towards the poles of the cell, and as they move, a spindle starts to form between them. And okay, so this is a diagram, and this is basically showing that the two white string are represented as chromosomes, and that's how they're getting split apart.
My name is Bryan Wynn, and I will be talking about the next phase of mitosis, which is metaphase. During metaphase, spindle fibers attach to the centromere of each pair of sister chromatids. The sister chromatids line up at the equator, or center, of the cell. The spindle fibers ensure that sister chromatids will separate and go to different daughter cells when the cell divides. Centromeres are what attach sister chromatids together.
The next stage of mitosis is anaphase. During anaphase, the sister chromatids separate and the centromeres divide. The sister chromatids are pulled apart by the shortening of the spindle fibers. One sister chromatid moves to one pole of the cell and the other one moves to the other pole. So at the end of anaphase, both poles have a complete set of chromosomes.
Alright, so the next phase of mitosis is telophase. And telophase is, I wrote down the definition over here, and we have a diagram coming up. And during telophase, the chromosomes begin to uncoil and form chromatin, and chromatin is uncoiled DNA. This prepares the genetic material for directing the metabolic activities of the new cells, and the spindle also breaks down and new nuclear membranes form.
Cytokinesis is the final stage of cell division in eukaryotes as well as prokaryotes, but it's not part of mitosis. During cytokinesis, the cytoplasm splits into two, and the cell divides. Cytokinesis occurs somewhat differently in plant and animal cells. In animal cells, the plasma membrane of the parent cell pinches inward along the cell's equator until two daughter cells form. In plant cells, a cell plate forms along the equator of the parent cell. Then, a new plasma membrane and cell wall form along each side of the cell plate. In our diagram, it's showing animal cells.
So I hope you enjoyed our presentation, and this is by Bishesh, Mrugank, Jason Garza, and Bryan Wynn, and we are in Mrs. Richardson's 7th period class. Thank you, and see you later.